due to technical problems, I was not able to upload this video, on the scheduled date and time. I have got a question for all of you. Okay, the objective I will tell you later, objective of this session. I have taught you about testicular tumors. Mostly that I concentrated on the etiology in the previous class, that was 2 weeks back. And Dr. Disha has taught you about testicular tumors in detail. So the question now is, you know about seminoma. We say it is the commonest malignant tumor of testis. But just remember, before I ask you the question, the suffix oma, I keep telling everybody. We have taught you that suffix oma denotes that it is a neoplasm, a tumor. And usually when we use the suffix oma, we say it is benign. But there are exceptions to this rule. Seminoma was a tumor which we were talking about. Seminoma is a highly malignant tumor. It's a germ cell tumor. Of course, it responds to radiotherapy, whatever. So now, what are the other tumors that you should remember? Lymphoma is another one. Lymphoma is also a malignant tumor, though we use the suffix oma there. So the question is, what is the female counterpart of seminoma? Okay, this answer should come to you. I'm sure all of my students know the answer. In other words, I can say, what is the ovarian counterpart of seminoma? Okay, look at this. I'm showing the picture of that also, microscopic appearance, and both these, the ovarian counterpart and the seminoma, appears microscopically similar. And even the suffix oma, I'm giving you half answer, is used for this female counterpart or ovarian counterpart also, even that is a germ cell tumor. What can you see here in this picture? 3 things are very clear. One is the tumor cells, which are here, which are looking so bluish. So they are arranged in, you can say, nest or lobules, because that group is separated now. So if there are a group of people and we put barricades separating the groups, so it's like that. So the stroma is separating the entire group of cells. So this is a stroma separating, and then it's separating them into lobules or groups or nest, whatever you can say. So this is the stroma. And what is there in the stroma? So there are tiny dark cells. So 3 things we are able to appreciate here. The tumor cells, the separating stroma, here it's slightly more, and the cells that are there in the stroma. So these are the tumor cells, this is the stroma infiltrated with lymphocytes. So did you get the answer? This is how it looks, the answer I am looking for, the female counterpart of seminoma. Usually it is unilateral, variable in size and encapsulated. So when I say female counterpart and usually unilateral, you should think of 2 things. But I am concentrating on the testicular tumor and a similar tumor in females. So counterpart of testis, you know what is there in the females. Okay, ovary. So we're talking of ovarian tumors. So it's usually unilaterally capsulated, and look at the outer surface. It is so smooth and basilated, and that surface is quite fleshy, solid, and you may say homogeneous. So the answer is dysgerminoma. Okay, so dysgerminoma is a malignant germ cell tumor that occurs in the ovary. The male counterpart of dysgerminoma is seminoma in the testis. So remember, germ cell tumors of testis and ovary, seminoma and dysgerminoma. Microscopically they have a similar appearance. So what is this specimen then? This is orchidectomy specimen. That's it. Why are you seeing these squarish windows there? That is where the tissue is taken by the pathologist for making slides. On the slides you can see the sections of varying sizes, right? Okay. So there is no normal testis that is identified here. I have told you, I have shown you also how normal testis appears and what is the string sign. The entire testis is replaced by a tumor here. Look at this. This is the upper end. So this is a cut section of the tumor. The testis is enlarged here, and at the periphery we can still make out that some normal testicular tissue, and this is the tumor part. There's lobules of tumor cells. So pink tan tumor, you can say. So homogeneous cut section, lobulated appearance. This is appreciated very well. Homogeneous cut section. So here again, this is the tumor. It appears so well defined, and this is the tunica, outer wall we can make out, and this is still the normal testicular tissue. So it's not invaded or occupied the entire testicular tissue. So here the tumor cells are in a mess. They're nice groups. So all these are the tumor cells, tiny lobules or groups of tumor cells. So in between is the separating stroma, and we can very well make out that there are nice lymphocytes here. So only three things that microscopically we want to appreciate. So the objective of today's session is only to make you draw a neat diagram of Seminoma testis, and once you're back we'll show you the real slide. So it's just virtual, the images that you are seeing, and then drawing a diagram of this. So uniform population, how are the cells? Uniform population of cells. They've got a very well defined cell membrane. It's very nice. That's why when the cell membrane is well defined, you can make out the entire shape of the cell. So for example, in a giant cell, say of tuberculosis, where the cells are merged together, we cannot make out the outer rim of the cytoplasm. And these cells have a very prominent nucleus. Of course, one of the characteristic feature of malignant cells is hyperchromatism. So here it's a prominent hyperchromatic nucleus. But however the chromatin, how is the chromatin arranged? So it is dispersed, so that once it is dispersed, we can appreciate the nucleoli inside this very well. So it's vesicular nucleus and prominent nucleoli. Tumor cells arranged in small nests and clusters here, separated by the stroma, and stromal lymphocytic infiltrate as very well seen here. Fibrous tuberculi or the fibrous tissue stroma separating and infiltrated with lymphocyte plasma cells. So what does it tell us? Any tumor that stroma which has a lot of lymphocytic infiltration, we say the tumor host. It talks about the host response to the tumor and has a better prognosis. So you should remember tumors which show this. One is this Germinoma, I already showed you. Then second is Seminoma. And third we should remember is another carcinoma of the breast, that is medullary carcinoma of the breast. Then even basal cell carcinoma. Characteristically they have lymphocytic infiltration in the stroma. And what else when the host immunity is good? So how does it respond? So how does the host respond to tubercle bacilli? By formation of granulomas, right? So even here we may see sometimes granulomas in the stroma and some giant cells also, syncytial type of giant cells. So this is a picture from the book you read. So very well defined cell membranes. Why am I stressing on this point is when you are writing a diagram, that's how the cells should be depicted. So lobular pattern, this is scanty stroma, lymphocytes, the prominent nucleolus here and a vesicular nucleus. So uniform cells, vesicular nucleus, prominent nucleolus, scanty stroma that is infiltrated with lymphocytes. So same histological picture you can see here. Occasionally eosinophils are there here. Yes. So I have already described the appearance of the cells. This is typical of a classic seminoma. So you know about anaplastic seminoma and seminoma that occurs in elderly, that is spermatocytic seminoma. where the cells are of varying sizes. Usually spermatocytic seminoma occurs in the elderly, and cut section has a mucoid or gelatinous appearance. Three things you should know about spermatocytic seminoma, because this was once a short answer question also. It occurs in elderly, and microscopically we see three types of cells, like they look like the spermatocytes in various stages of maturity. And cut section, here we said in a classic seminoma, it is grey-white homogeneous appearance. There the cut section is mucoid and has a gelatinous appearance. Why are the cells? I didn't tell you about the cytoplasm. I think these cells have a vacuolated cytoplasm. Why the cell not vacuolated, clear cytoplasm? Why the cytoplasm is clear? There is something which is there and the cytoplasm has dissolved. How did it dissolve? And what is it that is dissolved? So glycogen. These cells have, the cytoplasm has glycogen, and the glycogen has dissolved due to processing. Now when we remove the tissue outside, the glycogen dissociates into glucose, and what is a common fixative that we use? It is formalin. Formalin is made up of, it's an aqueous fixative. It's made up of formalin that is concentrated, one part which is mixed with 9 parts of water or saline. So it dissolves. That's why we see clear cytoplasm. And to say that these cells have glycogen, so we have to take the tissue bit, the biopsy, put it in a non-aqueous fixative, say about alcohol or any other fixative. So the glycogen remains, and then use a special stain. All of you know one of the special stain for fungus is PAS, for fungal demonstration, pyridic acid. And the same stain demonstrates glycogen also. So this is about seminoma, classic seminoma, and spermatocytic seminoma. I told you, 3 features. Grossly, both clinically and morphologically, it is distinctive from classic seminoma. 5% of all the germ cells constitute spermatocytic seminoma. It occurs in older patients, and tumor is bilateral in about 10% of patients. And spermatocytic seminoma, cut section, has a gelatinous appearance, and the tumor cells considerably vary in size. It is a very small lymphocyte-like to that is huge mononuclear cells, and stroma lacks lymphocytic infiltration, and it's got a better prognosis. When I say it's very slowly growing and rarely metastatizes. So the tumor is quite radiosensitive. Spermatocytic seminoma, please read about it. It is one of the types of seminoma, occurs in very elderly. 10% of cases may be bilateral. Then the tumor cells are of varying sizes. They are not uniform like this. Stroma lacks lymphocytic infiltration. Grossly, the cut section of the tumor has a gelatinous appearance. It's not homogeneous. Then it's got a better prognosis, we say, because it rarely metastatizes and it is radiosensitive. Even the classic seminoma is radiosensitive. Okay, let us draw the diagram of seminoma now. Seminoma testis. Okay, all of you need to write this, draw this diagram and we'll submit it. So what are the things that I have told you? Important is the cells are divided or arranged in lobules. So let us put a fibrous stroma now to show that therein lobules are there in nests or groups. Okay. Yes. So lobules, these are nests. What is this? This is stroma. How is this stroma? Stroma is made up of fibrous tissue. I am just showing fibrous tissue. So it has blood vessels, everything. Okay, this is fibrous tissue. Okay. Scanty fibrous stroma. Done. Actually the cells here are spindle shaped. If they are compressed, we may not be able to make out this. This is fine. So diagrammatic. So what is the stroma infiltrated with? So we are going to a secondary object now. So, lymphocytes. Lymphocyte, don't write it like dots. So they are nice round cells. So lymphocyte rich stroma. Okay, write the lymphocytes now. Okay, so two things are done. Now the composition, cellular composition. Is it uniform cells? Well defined cytoplasm, cytoplasmic margin or the cell membrane. Okay, so fill these with uniform large round to oval cells with well defined cytoplasmic margin. See, we are not depicting variation in size of the cells here, pleomorphism. We say one of the characteristic features of malignant cells, which is not seen here. Then we are not writing, leaving the cytoplasm clear because it is called glycogen. Now writing the nuclei which are vesicular, so opened up chromatin, vesicular nucleus, opened up chromatin, center, you can write them in the center. What else? When it is like vesicle, a drop of water, we can see what it contains. So the nucleoli to be written inside. Nucleoli. So that's it. You're going to fill the rest of the microscopic field with such tumor cells. So I'm just doing it. You're following me? Okay, the uniform cells first, then the nucleus and the nucleoli. So this diagram holds good both for this germinoma and seminoma. So uniform cells. Maybe it's easy for me to write. You may take some more time. The position of this blackboard, it's not easy for me to write. And, okay, I can't do it from this side because the right hand is. One or two nucleoli, I am just writing one nucleolus here. In a classroom, I make students write it. Help me in drawing this. Almost over. Let us label this diagram. So what is it? This is Seminoma testis. Why am I telling you to Seminoma testis? So this germ cell tumor can occur, similar tumor, wherever the germ cells are there. So midline germ cell tumors, starting from the pineal gland, apart from testis and ovary. So they are the sites for germ cell tumors. So these are lobules of tumor cells. This is stoma with lymphocytic infiltration. Lymphocytes, okay. These are tumor cells. So how are they? The uniform. So vesicular nucleus, prominent nucleoli, then clear cytoplasm. So if you are asked about seminoma in the examination, we say a picture is worth 1,000 words. Draw a neat diagram and write all this. Nobody will expect you to write an essay answer. So microscopy is done. Okay. So draw this diagram and then submit the assignment through email to us, and go back, open your books, textbook, whichever you read, and then read more about it simultaneously. Please open surgery book, textbook, and operative procedure, and know everything about testicular tumors. Okay. Thank you.